Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and very good day everyone. So welcome back to our SID3019 lecture. So today we're gonna continue on what we have missed from last week, okay? And then afterwards we'll see if we can finish it on time, okay? Now so first thing first, I would like you guys to actually do this, okay? So how's your understanding so far? If you think it's too difficult, if there's any concern, if there's any ways by which you want me to change the style of my lecture, do let me know. I'm always welcome for any feedback, okay? So for this one, as usual, you just open your Poll EV application and then type it in. I'll keep this online until week 6. So we will have 2 asynchronous lectures. So this will be the first one, and next week and the following week will be the second one. Alright, so I'll just keep this open, so any feedback are welcome. Just open your Poll EV and let me know. And do not worry, because this survey is anonymous, so I have no idea who you are. Alright, okay, so let's move into our lecture. So this is where we stopped last time, whereby we are looking at where we are looking at the differences between a traditional and a more modern approach. Okay, so we've touched about this. So I'm not gonna touch about it again. I'll just skip this, just to recall a little bit about this, about DNAs, okay, about mRNA, for example. mRNA, okay, and whereby I mentioned that mRNA is what Pfizer used for their COVID-19 vaccine. Okay, so we will look at that again today if we have time to actually view it. Otherwise, we'll cover about that particular section in the next week's lecture. Depending on if we actually have time, we will touch a little bit about the rapid test kit or RTK kit. Otherwise, I'll just skip that altogether. We will see. Okay. So this is what I missed last time. So immunity for a person comes in 2 pathways. This is a very common ones. Of course, if you go into very detail about immunology, then you might see there's more than 2 ways. But so far, just to make sure that it's easily understandable, it commonly has 2 pathways. So the first path is what we call as path that activates the natural killer cells, or NK cells. The second one is a path whereby antibodies are being produced. So this is what you've been heard multiple times, at least during this COVID-19 pandemic. There's a lot of talks about antibodies. So we're going to look at it today. So how does vaccination work? Okay, if you look at this, let's focus on that a little bit. So this is what we call as dendritic cells. So it's in our body. It's a part of what we call as white blood cells. So everybody knows white blood cells, hopefully. So dendritic cells is part of a subset of white blood cells. Okay, and as what white blood cells does, it protects your body against a foreign invasion, pathogens, viruses, bacteria, and so on and so forth. So this slide is to actually introduce you on how vaccination actually works. So, and you might notice there that it says a matured dendritic cells. So of course, if it's a matured, then there could be a younger self of that particular dendritic cells, which I'm not showing here. Okay, so what you know, because of course immunology is very complex. If you were to go into the very fine detail, then it's a bit complex. So I'll just straight away move into how vaccination works, and it starts off with matured dendritic cells. Okay, so we have one matured dendritic cells, or a cluster of matured dendritic cells. And then if you are looking at these 2 pathways, remember, we will look at more about T cell dependent and B cell T cell independent pathways. I'm not going to touch about it in very detail, but this is what will happen when a vaccine was delivered, or at least this is how our body reacts when there is a foreign object enters our body. Okay, first of is, normally you have two clusters of cells. So you have the matured dendritic cells on the left hand side. Then of course you will have the naive B cells. That's what we call it, the naive B cells on the right hand side. And once a bacteria or foreign objects enter our body, so what it does is it activates, or a component or part of the foreign object actually binds to a receptor. Again, receptor is a protein. Okay, so it has a very specific topological three-dimensional shapes. It's similar to a protein. Okay, but receptor is when a protein sticks on the surface of a cell. So we normally call it as a receptor, but it's still a protein nonetheless. Okay, so what happened was naive B cells, they come into contact, or they encounter these foreign objects via the receptor. Okay, and then what they do is they engulf. Of course, I'll have another slide talking about in more detail about this. So I'll just make it like very general for now. Okay, so they engulf. Engulf means that cells are flexible. It's not rigid or circular like what is shown in this lecture. Okay, so it's a bit flexible. So once it's flexible, what it does is it actually engulfs. It's going around the object. Okay, so imagine that's your antigen, or that's your foreign object. Okay, and this is your cell. So initially the cell comes into contact with the foreign object, and then what it does next is it engulfs the foreign object. Okay, it's kind of like eating up the foreign object. And then what it does, it completes a process called antigen processing. Sorry, antigen processing over there. So okay, so what happened next is that the foreign object is being processed, and then it will be represented on the surface of that particular cell. So over here, you can see, if you notice, is that there's a triangle shape on the foreign object. Okay, and then after antigen processing, the red color object is now being presented on the surface of the B cells, and the name changes from a naive to a mature cells. Okay, so that's the process of antigen processing. We will have a slide on it soon, but this is just an overview. And then at the same time, the foreign object was also identified by the matured dendritic cells. Of course, before matured, you have a naive dendritic cells, and naive dendritic cells also accounted similar. Okay, so the idea here is similar foreign object. So now you have a matured B cells and a matured dendritic cells, by which the dendritic cells activates a naive T cells. Oops. Activates a naive T cells, and this naive T cells then becomes a prime T cell. So it's not a matured T cells, they call it as prime T cells. And then once these prime T cells and this matured T cells comes into contact, they share signals in a T cell dependent. So that's why we call it T cell dependent, meaning that without T cells, these matured B cells won't do anything. They will just stay still without any signal. So but when a T cells activated T cells or prime T cell comes into contact with these matured B cells, they share the information. And then what happened is that the B cell now diversifies. So it produces either a memory B cells or a plasma cells. So a memory B cells is actually kind of like how, well, the word memory should tells you something. It memorizes. So it memorizes this particular antigen there. Okay, so that in the future, if the memory cells encounter similar foreign object, it can straight away, quickly produce plasma cells. And the function of plasma cells is voila, producing antibodies. Okay, so this is the pathway from a foreign object, bacteria or viruses, until the production of an antibodies. Of course, this is a simplified version. There's a lot of things actually comes to contact. Even the receptors, there are multiple receptors that are actually talking to one another before you can actually produce an antibody. So this is a summary. Okay, and the word secondary infection here is very critical because this is how vaccine works. Okay, you have a vaccine, a first dose or second dose, it doesn't matter. It's a vaccine, a synthetically produced foreign object. Okay, and then you want your body to actually produces a lot of these memory B cells, so that if a secondary infection that comes from the real source, the bacteria or the in our case nowadays is the COVID-19 virus, it very quickly activates the plasma cells, thus producing antibodies to help our body to trap or deactivate or neutralize the COVID-19 virus. Okay, so this is the path of a T-cell dependent. T-cell dependent path is the only path that produces good memory B cells. A T-cell independent pathway, on the other hand, is just straight away activates. Of course, it involves antigen processing. Okay, because without processing, you cannot present. Okay, without presentation, you cannot produce antibodies. So from a naive B cells over up here, okay, when they encounter a clustering of molecules that mimics the foreign object or similar foreign object. For example, this can be a synthetic vaccine. Okay, so chemically synthesized synthetic vaccine, for example. Okay, you can still activate the B cells, but it doesn't guarantee that you are actually activating the memory B cells. So you can still produce antibody, you can still get a protection, but the protection will not last long. Okay, so this is a very simplified version of immunity. So it's not just about vaccine, it's about immunity. And we will go into a bit more detail on looking at how vaccine actually works. Alright, so I've talked about antibodies. So what are actually the antibodies? So antibodies, if you look at the structure, you should straight away say, oh, it's a protein. Okay, so there's a huge structure, molecular structure, and if you notice there's a lot of SS bond shown in the picture. So this SS bond is actually what we call as the disulfide bond. Okay, so disulfide bond, again, it's a how do I say this? It's a type of forces inside a protein. Okay, so this is what antibodies generally look at. Alright, so antibodies are being produced by B cells. Of course, they are. It's almost similar. I'm not going to say that it's identical. So it's similar to a protein receptor on the surface of B cell. So you'll remember in the previous slide, I was saying that there is a presentation, so a processing, and then the foreign object actually being presented outside of the cell. So the protein that actually presents this has almost the same structure as the antibody itself, so that you can actually say, if you actually extract out this protein that represents the foreign object, you purify, you remove the object presentation, there is a very, very high chance that this similar protein can also bind in a similar way as antibodies. Okay, so again, it's similar, it's not identical. The size of an antibody is roughly around 14 nanometer across, 8 nanometer in length, and about 4 nanometer in depth. Okay, so if you imagine, if you are looking at it from a sideways, if this is a Y shape, okay, so if this is a Y shape, all right, for the antibodies, if you move it sideways, so this is about 4 nanometer long, and then you have 14. I think this one should be the 8 nanometer, and then 14 nanometers in length. Okay, so and there are many types of antibodies. So antibodies, if you recognize it in a biological format, It's normally start with a big I and a G, and then a letter next to it. So there's IgA, as it says here, IgG, which is systemic antibody. Systemic. Okay, systemic antibody. This is what the antibodies that are normally can be found throughout our body. So regardless of if you take blood from your finger, for example, or you extract out, you can definitely get an IgG. If you take it from your brain, if you take it from your ears, if you take it from your legs or whatever, if you purify it, you can definitely get an IgG as part of the antibody component. And then you can also have an IgM, an IgE. Of course, they have their own functions, which will not be discussed over this slide particularly, and we will talk about it in the next slide. Now, something that you need to know about proteins, antibody proteins, is that there are this antigen binding site, okay, at the very end of the Y shape, and that if you can see here, you have two two components of the protein, okay, so which are connected just by a disulfide bond. So the antibody is actually not a single polypeptide chain. So this antibody, you can consider it as the fourth level or a tier. It is a quaternary structure of a protein. Okay, so you have one peptide sequence, peptide chain over there, and of course, if you look at all of these in the middle, okay, again, it's just connected by disulfide bond. So that one is a second polypeptide chain. And then similarly, you have a third polypeptide chain and a fourth polypeptide chain. So antibodies are actually made out of 4 polypeptide chain. So it's a definite answer of a quaternary structure. Okay. The shorter version, we call it as a light chain. The bigger version is what we call as a heavy chain. These are... Why do I want to talk about antibodies? Because these antibodies are related to the technology that is being used in many rapid test kit. Okay, so that's why I want to go in a bit more detail about it, so that when we go into the RTK lecture, if you actually have time to actually talk about it, then at least you will know a little bit information about this is why this is this and this is that. All right. Now, so those are the 4 critical things about antibodies. Of course, I can always talk about it in more detail, but due to restriction in time, I will just stop there. Okay, so those are the main critical thing that you need to know. Right, so this is just a figure showing you about how antibodies bind to a COVID-19 virus. So, of course, this is not a real image because COVID-19 viruses is about 12 to 16 nanometer long. Okay, and as I mentioned, the length of these between the Y chains is roughly about 8 nanometer long. Okay, so this picture is slightly smaller than the real one, so but it's still acceptable. So this is how antibody works. It binds to your target. Okay, so you have this the Y shape, the heavy and light chain at the very end. That is the binding site for that particular foreign object. In this case, it's the crown of the COVID-19. Okay, it binds to that particular crown, go around it. Again, if you recall, SID3020, we're talking about proteins and enzymes, for example. So it has a similar function, but enzymes catalyze some reaction. This one is not. It just goes around and bind it. But there is still a flexibility. There is still a dynamic modes of functioning construct of the proteins going around the target. Okay, and then this region at the back of the Y-shaped antibody is where we call it as an FC region, and this region can be identified by different types of white blood cells, and therefore signals the white blood cells that, oh, I'm attaching my body into one foreign object, so come and eat me and process me. This is a bad thing. So white blood cells will come and again engulf around the objects, taking it inside the body and then kill it. Okay, so that's how generally how antibodies and our immune system works. Alright, so this slide is just to show you the differences between the types of antibodies. So you can see here on the far left, you do have an IgG, they call it IgG pentamer, IgM pentamer. So that one is IgM, that's IgG, which is the most abundant in our body. You have IgA, a secretory IgA, so it's mostly in our lungs areas, our mouth, up until our trachea. We have IgE, so this is a signal on mostly on allergic response, and you also have an IgD. Okay, so if you go through down a little bit, you can see that the differences between all these antibodies are part of it, the number of antigen binding site. So IgM, you have 10 binding sites. Okay, again, if you count that from the previous slide, so you have 10 of it. Okay, IgG, you have 2. IgA, because it's kind of like two IgG close to one another, even though it's not exactly like that, it's similar. Okay, you have 4 binding sites. IgE, you have 2 binding sites, and IgD, you have 2 binding sites. Okay, you can look at the weight of each antibodies. Okay, so 900,000 Dalton for IgM, around 150,000 for IgG. IgA is around 400,000, 200,000, and so on and so forth. So you can read through all of these. And most important thing is that you can see the percentage of total antibodies in the serum. Serum means that it's in the blood. Okay, so in the blood, the most abundant is IgG. So that's why we normally call IgG as a systemic antibody. And if you were able to produce this IgG using your vaccines, for example, you will have a higher chance to produce a protection against the foreign object of interest. Alright, so and additionally, you might also heard previously about why mother's milk is the best. So this is the reason. Okay, so for those who actually for the babies who actually being breastfed, IgG actually cross the placenta. That's one thing. So the antibodies can actually cross placenta during pregnancy. And the second one, IgG actually enters milk, and then when the baby feels fit on the milk, then the antibodies goes into the baby's body. So it's an extra protection for the baby. Okay, so Fc region binds to. So this is the Fc region, the Y, the stem of that particular antibody that I mentioned. So it binds to phagocytes. Phagocytes is another type of white blood cells. Phagocytes is cells that engulf or eats out, phagocytize, okay, a foreign object. So IgG promotes these white blood cells to eat the target. Okay, and this one is IgE. It's a mast cells and basophil cells antibody for allergy. So it's identify allergic component, and also it has antiparasitic activity. Okay, so it involves antiparasitic. Alright, so that's very brief about different types of antibodies. Again, I want to talk about antibodies in a bit more detail, because if time permits, then of course we're going to talk about how this technology can be used for RTK, okay, rapid test kit. Okay, so this is a microphage, or a type of phagocyte. Okay, this acting of a cell going around the target is called phagocytosis. Okay, phagocytosis. So that's why you call it as phagocyte. Cyte means a cell. Phago reflects on the phagocytosis, the process of eating out. And one of the cells that does this is what we call as a microphage. Okay, so on the surface of this microphage, there's a lot of this Fc receptor. Again, we are talking about IgG. Okay, so there's a lot of Fc receptor on the surface of this phagocyte. So once the antibody binds to a pathogen or the foreign object, it can signal on the phagocyte, or I have something attached to me, so come and eat me so that you can clear this thing out. So this is what the cell is doing, is going around the pathogen. Okay, so this one is a true 2D structure, but if you think about, you know, a 3D type structure, so if you have a foreign object over here, let me see. Okay, if you have a foreign object like this, so instead just going around this way and that way, it's actually going around in a whole area. It's like going around like this and then eating up the foreign object. Okay, so that's what it does, and that is actually what I've planned for last week's lecture. Unfortunately, we don't have time, so I didn't manage to go through everything. But yes, I'll just continue with today's lecture, a bit more information about vaccines. Okay, so references, you can always do that, and all of these information are actually part of my thesis for my PhD. So this is what I did for my PhD, and of course, you can always refer to those internet resources to have more reading about that particular topic. Alright, so attendance. Today's code is asynchronous lecture via Spectrum, and if you actually QR code this, you will find some nice information. Okay, do so if you want to. Okay, so part 2 all about vaccination is the related technology. Today we're just going to focus on vaccines because I'm sure I don't have enough time to actually complete everything, and hopefully next week I can touch a little bit, I can finalize about vaccines, touch a little bit about a rapid test kit as well, and then the following week we will have a tutorial and some discussion, and the week after that we'll have a quiz. Okay, let's move into it. Number 1, we'll talk about vaccines. So if you understand immunology, if you understand what I have been talking about for the past 2 weeks, and just now, so for a vaccine to work, there are 2 components that are required. Okay, and if you're answering B cells and T cells, that is true in some sense, but that is not the 2 components that I'm talking about. Okay, so the 2 components that are required are the antigen. Okay, the antigen is the foreign object, so we'll talk about that soon. Okay, and another one is a danger signal. Okay, so a danger signal. So a danger signal alerts the body saying that something is wrong. So please, white blood cells, come by, have a look. If there's any issues, then please solve them. Okay, so this is what danger signals are all about. So danger signals can come in a wide variety of objects. We will talk about it, a few of them, or probably 1 or 2 soon. Okay. So and of course, when we're talking about vaccines, there's always a traditional vaccine and the modern vaccine, as what I've mentioned in the early of this lecture. So please recall what are the differences. Okay, put it in. I will have a question over here. So it's kind of like our pop quiz. So normally I'll use a poll EV. Okay, poll everywhere, but because this is an asynchronous lecture, I cannot do that. So instead, I'm asking you guys to watch Spectrum. Watch this video using Spectrum, because there will be an embedded quiz over here for you to practice your understanding on the concepts that have been covered so far. Alright, so let's move on to vaccine technology. So of course, traditional vaccine is, as what Edward Jenner did, is the use of whole pathogen. Okay, nowadays we are still using part of what traditional vaccines are, for example BCGs, Okay, even at least 7 years ago when my son was born, doctors still injects with BCG, and as far as I know, BCG is still used in Malaysia. Okay, so BCG is why do I call it as a traditional vaccine? Is because BCG still uses a whole bacteria. So they are using a bovine tuberculosis bacillus. So bacillus is a type of bacteria. Okay, bovine means a cow or its species, cows and goats and whatnot. So it calls as bovine. Tuberculosis bacillus is the type of a disease. So tuberculosis, if you have heard, it's TB. That's what we call it, TB. Okay, tuberculosis, but in Malaysia we in bahasa we call it as TB. So recall Edward Jenner. What is the story behind how Edward Jenner produces vaccines against smallpox? So I will probably ask that in the quiz. Do remember, okay? And the picture on the right hand side is just showing you on how BCG actually acts. So you know, that one is the sample of BCG vaccines, and this is how BCG vaccines related to the different malaria diseases, but you know, just an image. All right. So modern vaccines, on the other hand, is using part of a pathogen. Okay, so part of a pathogen, or bacterial, biotechnological products formulated for enhanced efficacy. Okay, the keyword here is formulation. Formulated is part of the danger signal that I mentioned initially. Okay, it's part of a danger signal, so it's a formulation. So how you formulate it, then we'll talk about it soon. But the figure on the right is showing you what a modern vaccine is. So you can see on the left hand side, they are using a plasmid DNA, okay, plus a liposome. So liposome is a formulation. It's also a carrier molecule, and so on and so forth. So you actually incorporate the plasmid, the vaccine component itself, into a liposome, forming this structure. Okay, you might have noticed that, oh, that structure looks familiar to something that I've seen. We will go through that soon. Okay, or additionally, you can also use mRNA, which is a messenger RNA. I hope that you guys have gone through SID3019, where my colleague have talked about nucleotides. Okay, nucleotides including DNA and RNA. So hopefully you now have a bit more understanding on why do you need to learn all of these. Okay, so you have a plasmid DNA over on the left hand side, you have mRNA on the right hand side. Again, you can have a carrier, a similar carrier, a liposome. You incorporate the RNAs, the mRNA inside the liposome, and then you deliver it. Okay, you deliver it as a vaccine. It goes into the cells. Okay, it's being processed either in the nucleus for DNAs or for mRNA. The processing is just happening on the cytosols or the components of the cell. So if this is a cell, okay, that is the nucleus. So cytosol is over here. Okay, everything over here, we call it a cytosol. All right. So mRNA is just being processed there, transferred to ER complex, endoplasmic reticulum, and being produced as a protein, okay, or protein or peptides. And then this protein or peptides, because they are foreign, so we call it as antigen, and then will be presented on the surface of a cell, and thus this is how COVID vaccine works, in a very similar concept. You might not understand the whole thing yet, so we will go to the next slide to try and give you more understanding on what is the technology behind vaccines. Okay, so what happened when a vaccine is injected into your body? So you normally see a picture of a nurse carrying a syringe and then injecting on the person. Okay, so what happened there? So when the vaccine is being injected in the body, so imagine that this is your skin. Okay, just imagine. Hopefully you can imagine that that particular line over there is the skin. Okay. So when you inject, before I go there, so I'll probably just finish up what I've written here. So the part for antibody production or any protection, the immunity protection, so to say, is first identification. Okay. Second one, processing, and the third one is presentation. Okay. So the foreign objects or the vaccine construct, the vaccine itself, not the syringe. So the syringe is being used to inject the vaccine. So the vaccine itself needs to go through 3 processes before antibody can be produced. Okay, identification, processing, and presentation. Now, so when you inject it there, okay, on the surface of the skin, so that's why I said site of an injection. Okay, a phagocyte, such as antigen or different names is a different class of white blood cells. We call it as antigen presenting cells. If you don't know what antigen is, wait for a while, then I will touch about antigen soon. Okay, so we have this group of cells called as antigen presenting cells. So they will come to the site of injection, normally because of the pain or the signal from the vaccine itself that attracts these antigen presenting cells. So it goes to the site of injection and it counts the molecule of that vaccine. Okay, imagine, so I've drawn it like that. So one is antigen, one is adjuvant. So we'll talk about it soon. What is antigen, what is adjuvant? But imagine this is the liposome that you saw in the previous slide. Okay, imagine that one is the liposome, and then you have your mRNA or DNA in the middle of the liposome. Imagine this. So imagine that one is a circular complex. You can imagine it's the green one or the yellow one. It's fine. So what it does is the antigen presenting cells actually engulf or phagocytosize this particular molecule into its body. Okay, and then what happened next is what we call as that one is site of injection. That's step number 1. Step number 2 is phagocytosis or pinocytosis. You can always look up the terminology. And then number 3 is antigen processing. So this antigen processing is part of a critical component whereby the antigen itself is being processed inside the cells. So in case for mRNA, of course, there's a lot of things going on. The mRNA produces protein first, and then the proteins is being processed before, and towards the end of the day, it will be presented outside of the cell, outside of the antigen presenting cells, in a major histocompatibility complex, or MHC, type 2 or type 1. I have a figure to describe about this soon. Okay, or additionally, you do have alternative pathway to actually enter the APC, antigen presenting cells, is via receptor-mediated endocytosis. So this particular component binds to a receptor that is available on the surface of this APC and then enters the cells. So an example of a receptor, the type of receptor is TLR, which is toll-like receptor. I also have a slide on this particular receptors soon, or not really a slide, more like a brief on it. Okay, so this is how generally antigen processing works. And then after it's been presented, either in MHC class 1 or class 2, so you can have either humoral response or a cellular response. The difference is a humoral response is more on throughout the body, it's outside of a cell. A cellular response is more on activating cells. So you can see the NK cells, natural killer cells, I mentioned in the first slide for this particular section. You have CTL, cytotoxic T lymphocytes, and also you have a macrophage. Okay, so you activate these cells. When it means by activation is that a signal will come to these particular cells and ask them multiply. So we have been invaded, protect the body, so to say. Okay, so it will multiply, and then because from the first slide I've shown, there are receptors on the surface of these cells, so thus the immunity system will kicks in. Okay, but you can see that there is actually the path is actually very long. It's not as simple as you inject it once, you inject a vaccine, and then straight away you produce antibodies. It doesn't work that way. Okay, so there's a lot of research going on into producing a vaccine. You need to go through all these barriers. If one of the barriers is stopped, then that's it. It will not happen. For example, if antigen that you the foreign object that you actually put into the vaccine does not work, then you will not produce any antibodies. If you can see at the bottom here. Okay, so I've talked about cellular response. What happened in a humoral response? So in a humoral response, I'm not going to touch about this TH2, TH1 pathway, but what it means towards the end of the day is it activates B cells, and then these B cells produce antibodies that are either neutralizing or opsonizing. So what are the difference? So neutralizing means that it's just neutralized. So say for example, you inject in insulin, just for example, probably not insulin, probably an enzyme that can degrade your protein in the body, for example. Okay, so this is just theoretical. So you inject it in, but then your body has antibodies against that particular enzyme. So what it does, it stops the enzyme from functioning. Okay, so the act of stopping that particular function is called as neutralizing. So it neutralizes the function. Opsonizing, on the other hand, is to kill. Okay, so for example, a bacteria goes into your body. So if this bacteria, if the antibodies that comes into contact with this bacteria is just neutralizing, means that the bacteria will still be alive. Okay, so it just binds to that particular bacteria and nothing happened. Well, not to say nothing happened, but nothing happened to the bacteria. So meaning that the bacteria is still alive until the FC region of that particular antibodies signals the macrophage to actually come in and eat up the bacteria. Okay, so that is neutralizing. Opsonizing on the other hand is you have a bacteria, and when this antibody binds to the bacteria, it can promote the bacteria to kill itself. Okay, either it kills itself or it produces something that towards the end of the day the bacteria will be killed. Okay, will either explodes or whatnot, but we call it as opsonizing. So you can look up for that particular terminology. So we have 5 more minutes. I'll just quickly go through, and then if we can, then we will skip. But this is just another detail of antigen processing. This is the real pathway if you want to have a look. Okay, so this is the real pathway, and towards the end of the day, you can see there is a class 2 MHC complex or class 1. So this is the major histocompatibility complex that I mentioned. So there's class 2 and class 1. You can always go back to the previous slide to actually look at it. Okay, and this will not be covered in the quiz or exam. So this is just a particular information about processing, how antigen processing works. This particular slide will not be in, but anything that I mentioned prior to this slide can be asked in the quiz or tutorial or the exam. Okay, so this is just an additional info for those who might be interested in the process of producing antibodies, because towards the end of the day, every one of us actually have antibodies, and we has this process going on into our body without us noticing it. Okay, I think this is all for the first asynchronous lecture. I will continue next week talking about a little bit more detail about vaccine technology, going into some examples of vaccines that are currently on the market, especially with primary focus on COVID-19. And then if time permits, then I will talk about the RTK kit. Otherwise, RTK kit will be covered in the next week lecture, plus the tutorial and discussion. Okay, I think that is all for today. Thank you for listening, and I'll hope to see you next week.